internal magnetic forces.
Hi, my name is Kennedy Hunter, and today I will be presenting my experimental research paper. For my paper, I chose to look at how macroinvertebrates can be used as bioindicators of dissolved oxygen levels in water. Macroinvertebrates are aquatic organisms that lack backbones and are visible to the naked eye. On a hierarchical scale, these organisms fail to be recognized in aquatic ecosystems and are most often than not ignored entirely. However, when it comes to the water quality testing, these low-level insects are regarded among the scientific community as being one of, if not the most, reliable bioindicators. Given the fact that they spend much of their life in water, they are able to detect and report long-term and short-term changes in dissolved oxygen levels, which are just as they sound, the level of free, non-compound oxygen present in water. Dissolved oxygen is essential for the survival of fish and other aquatic organisms and can range anywhere from 1 to 20 milligrams per liter. Regarding macroinvertebrates, these creatures tend to fall on the lower end of the spectrum for dissolved oxygen requirements, needing only 1 to 6 milligrams per liter. In spite of this, they are still regarded as sufficient indicators, as levels that fall below this range are typically indicative of harmful water sources. A study conducted in 2008, which looked at how macroinvertebrates could be used as indicators of water quality in urban streams, reported that values that typically fell below the normal range were reported to have been harmful to aquatic fauna. This study also noted that factors that may have had a negative influence on dissolved oxygen included a rise in temperature or salinity, which is the amount of salt concentration found in water. Opposingly, positive factors included an increase in pressure in addition to the undergoing of photosynthesis. For this experiment, an environmental integrity index, which was a communal database, was used by lab groups on various sites of Waller Creek. Our first test, which consisted of using a YSI multiprobe in different sections of the creek, recorded both pH, conductivity, and temperature. Replicate measurements were documented at least 3 times in each of the following locations, and afterwards, the data recorded from each group was input into the communal database. To measure dissolved oxygen, nitrates, and phosphates, the V2000 photometer was utilized. For the DO analysis, a water sample was collected from the creek, and the photometer was turned on, and the program 141 was entered, which was specifically for dissolved oxygen testing. Before inputting the water sample, a blank vial was input and zeroed out to get a blank reading. The sample cup included in the water chemistry pack was filled up to the 25 milliliter mark with creek water. A DO vacuum vial, which contained a buffered yellow-green dye, was placed tip first into the sample cup and then snapped so that the water sample filled the vial. The vial was then inverted multiple times, subsequently mixing the water sample with the vial's original contents, which turned it a blue color. This blue color was representative of the amount of DO in the collected water sample. A chem wipe was then used to dry the outside of the vial, and it was then inserted into the V2000 photometer. 2 minutes later, a DO reading was given in ppm or milligrams per liter. The reading was then repeated for a second time, and if they were dissimilar, a third analysis took place. All values were then recorded and put into the class-wide EII database. The next sample collection, involving macroinvertebrates, took place back in the lab, following the conclusion of all water chemistry testing. Subsamples were collected from each of the respective lab groups' locations and were then poured into white trays. After being inspected in 3 sweeps by different members each time, the macroinvertebrates were extracted from the tray and preserved in bottles of ethanol. A dissecting microscope along with a reference collection was provided and used to identify the organisms in each sample. Following this observatory period, the data was also input into the spreadsheet, and a summary table of each group's findings were also provided. From the spreadsheet, I gathered a total of 29 data points, 25 of which had not observed macroinvertebrates. The average and standard deviations of both sets were first computed. For my smaller set of data, which observes macroinvertebrates, the average was 8.0965 milligrams per liter. The standard deviation was 0.65682. My larger data set, which did not observe macroinvertebrates, had an average of 6.10668 milligrams per liter and a standard deviation of 3.215864. With these points, I was also able to conduct a t-test, where the first distribution input was DL measurements with no macroinvertebrate observations. The second was with macroinvertebrate observations. Tails 1 was used because of the previously prediction that higher DO levels would present more organisms, and type 3 was selected because of the disparity between each distribution sample size. The resulting p-value was 0.00542, and when subtracted from 1 and multiplied by 100, it equaled a confidence of 99.457782%. Table 1 provides an overview of the calculations performed for the two datasets, including the average and standard deviation of each, along with the results of my t-test. Table 2 provides a visual of all the data points collected in the form of a scatter plot graph. It compares the DO levels recorded to if there was an observation of macroinvertebrates. From this representation, it can be interpreted that many of the data points with no macroinvertebrate observations were below a DO level of 7.0 milligrams per liter, which was less than all 4 of the data points collected that had macroinvertebrates. In regards to my central question, which looked to answer if macroinvertebrates could be used as bioindicators to detect dissolved oxygen in water, the data set with macroinvertebrates having a considerably larger average DO supported my first hypothesis that more macroinvertebrates would be observed within locations with higher dissolved oxygen. In addition, because the resulting p-value was lower than the standard alpha value of 0.05, there was enough evidence to prove the null hypothesis wrong and prove that there was a statistically significant difference between the two distribution sets. This further supported my second hypothesis, that lower DO levels correlated with the lack of macroinvertebrates. These results were to be expected, as macroinvertebrates were one of the few organisms that required little to almost none DO. Therefore, the locations that recorded levels below the accepted values were expected to not have any of these particular organisms in their subsamples. The conclusions formed and the calculations observed were also similar to the findings that I described earlier from the study conducted in 2008. However, I must note that although the data provided to me was accurate, it was not wholly reliable, as many of the contributors did not record all of their DO values or macrovertebrate observations. Additionally, the large difference in sample sets between both distributions is also not favored to perform a t-test. If more data points had been available for the DO levels with macroinvertebrates, the significance of the p-value calculated would be much more prominent and acceptable. Besides that, I do believe that this data is relevant, as it has proven to be an alternative form of water quality testing, which could help preserve time and money for many environmental scientists and groups. The presence of these organisms could also be used to identify pollutants in water. More simply put, macroinvertebrate collecting is an inexpensive and swifter process that produces the same results as a multi-step water chemistry assessment. In regards to future macroinvertebrate usage, a possible experiment that could be performed could use macroinvertebrates in a certain area of Waller Creek, and with the help of biological monitoring from the larval to adult stage, we would be able to note the changes in the organisms observed along with the related changes in the creek's water chemistry. For example, we would look at pH, conductivity, and dissolved oxygen. That concludes my presentation. If you have any questions, please comment down below and let me know what you think. Have a great day.